Hi, everybody. Sorry, we're starting a few minutes late here. There's always technical issues when we do things like this, and we had a few right before this webinar started. So I'll just give it another maybe 30 seconds or so. It seems like everybody's joining. And then we will jump right in. So we have a good group today. So I just want to start by introducing this webinar series and myself and my fellow co-host. So I am Kara McCreary, Greenhouse Vegetable IPM Specialist with the Ontario Ministry of Agriculture, Food and Agribusiness. So for those of you who haven't heard yet, our ministry name changed. So instead of being OMAFA, as we've been known for many decades probably, we are now OMAFA. So we lost the R, but we gained agribusiness. Anyhow, this webinar series is called the GrowON webinar series, and it's brought to you by the greenhouse team at OMAFA. So the greenhouse team consists of two greenhouse vegetable specialists, so Fadi Al Daoud, who covers production, and myself, who covers IPM. And then our counterparts in floriculture, Abby Weisner and Dr. Sarah Jandrisic. So I'll let Sarah introduce herself.
Yeah, hi everyone. Looks like we have a good group going, like Kara said. So just a few housekeeping things. When you've got questions at the end of this webinar or as you think of them, you can enter them into the Q&A function. It really helps us out if you put it in there. If you are having technical difficulties or you can't, you want to say great job or that's exciting, then put that in the chat. Oh, and polls. I knew it. I was about to forget. Every time I do a webinar, I forget to run a poll. So we want to know more about you guys. So we created a little poll that I'm going to launch. So if you can take a second and answer the questions that should be on your screen, we basically just want to know what your role is in the greenhouse and what crops you grow, so we know who's attending, and that can maybe help us answer questions at the end or things like that. So it looks like, I kind of love these real-time things. So it looks like we have a lot of growers or IPM staff, about 63%, a little bit of industry, and then some academia, government, and then other. And then what does your farm grow? It looks like we have a pretty good split between greenhouse vegetables. Oh, flowers is winning now. And then some nursery crops and a little bit of other. So I think that's where we're at. Yeah, greenhouse flowers won by 1%. So excellent. Okay, thank you very much for answering that poll. And now I'm going to turn it back to Kara to introduce our speaker.
Nice. Congratulations, Flowers, for coming in the lead. So yes, Sarah and I am going to speak for Sarah. I think she would agree with me here. We are really excited about our speaker today. She's been a long time, we've been in admiration of her for a long time. I think even before we started our careers with OMAFA. You agree, Sarah?
100%. I think a lot of us literally are in our careers because Hannah came to Guelph and presented, and we were all like, we want to be her.
Yeah. So we're still a little bit starstruck, but we got to work with her now. I hope she's blushing a little bit before she starts this. Anyways, it's my pleasure to introduce Hannah Fraser. So Hannah is the entomologist for horticulture crops at OMAFA, and she's been doing that job since 2000. So she has a lot of experience and a lot of experience with a wide variety of crops. So it's pretty exciting for us. She works as a crop specialist to develop pest management recommendations for horticulture crops. Hannah's adventures with resistant insects began as an undergraduate when she tirelessly reared predatory stink bugs for augmentative release as part of an integrated pest management program for Colorado potato beetle, a pest notorious for its capacity to develop resistance. Upon completing her graduate studies at the University of Guelph, she says a really long time ago, she worked with Agriculture and Agri-Food Canada to assess mating disruption technologies for management of orchard and vineyard pests. One of these pests, the oriental fruit moth, have become resistant to available insecticides, hence the need to develop alternative approaches to management. Her current focus is preparedness and response planning for invasive and emerging pests. So I'm happy to hand it over to Hannah now, and I hope you will enjoy the wealth of information she's going to share with us today.
Okay, great. I am going to stop my video and I am going to share my screen. So you'll let me know if we run into any issues. Okay, how's our view? Shall we switch it from speaker mode?
Yeah.
I need to hear from you. Okay.
Yeah.
All right. There we go.
There, looks good.
Excellent. Well, welcome everybody. I'm actually really excited to be here today. I've been watching some of the GrowON videos, like the webinar series, and I've been really impressed with them. And I kind of over the last week probably looked at half a dozen of them, and I think they're fantastic. And I need to kind of go back in time and look at them because it's there's a lot of great information. And I think what my goal was today was to kind of take some of the information that you've heard, because we hear about resistance and resistance management in all sorts of talks that have been presented through this series. And my hope is to kind of bring it all together. So hopefully that I can do that today, and there'll be some opportunities for us towards the end to sort of maybe ask some questions, maybe brainstorm a little bit, because I know that there's a lot of sort of unanswered questions that we have that are going on out there. So I wanted to start with my talk today with an agenda of what I'm talking about and what I'm not going to be covering. Although I realize when I say things I'm not covering, the reality is I probably am covering them. I'm just not covering them as sort of a focal point. But there's a lot of information that I think you'll see that is little tidbits that you can integrate into different parts of your management programs. So what I am going to cover today is provide that resistance 101. So what are some of the general concepts that are foundational for developing a resistance management program? And of course, resistance management is part of your IPM program. They are really tied together. So yes, the focus today is on IRM tactics for greenhouse, but the reality is that pretty much anything that I say today is going to be relevant for your production if you're a producer that's producing crops outdoors as well as in protected culture. And a lot of the information that I'm covering today is from or based on information that comes from the Insecticide Resistance Action Committee group, or IRAC. So hopefully people who are attending today know that IRAC has a really great website where you can go for information on resistance management. There's fantastic slide decks that talk about how insecticides work or how to develop resistance management programs. There's some great posters. There's information about the status of resistance for certain pests. So really, really fantastic resource. So what I'm not going to be covering today are sort of those other IPM best practices you're already doing. I'm kind of making the assumption that you're all familiar with how important it is to start out clean, to monitor, to use other tactics like cultural controls, in addition to the pesticides that you're applying in your program. So there's that kind of, I'm assuming that you're doing that already. I'm not going to be talking about products for specific pests or detailed rotation programs, although I will talk a little bit about what a rotation program would look like, or maybe provide some ideas on how to get one going. And I'm not going to be talking about compatibility with bioagents, but there is a lot of information that I am going to cover today. And I wanted to draw your attention to the picture on the left-hand side. So you will see some familiar foes that are there. There's whitefly, there's two-spotted spider mite, and there's an aphid there. And the reason that I think it's important to show these to you today is these are pests that are common in greenhouse production. And as it turns out, they're really important. They're significant pests globally from a resistance standpoint. So if you look at databases with these incidences of of resistance, you'll see that these pests have ranks number 1, 3, and 4 globally in terms of the number of cases of resistance as well as the active ingredients that they're resistant to. And I know that resistance management is really challenging in field production, but it's a lot more challenging in greenhouse production. I know you already know that. I was reminded of it when I was putting this talk together. So one of the things I think it's really important to talk about is that pesticides are they're a valuable resource, and they're actually a non-renewable resource, and we need to think about them with that mindset when we're using them. Some of the issues when you run into resistance is obviously that crop loss. You know, you've gone through, you've set up your program, you've brought your cuttings in, you've dipped them, you've got your bio program going, and you're moving along, and you're monitoring, and you know it's time to apply a product, and you grab a product that's worked for you in the past, and now it's not working. So now you've already invested all of that time and money, and you're running into a situation where things are just not working the way you had anticipated. That's a problem. There's also the whole thing about losing your flexibility. I mean, obviously having more good quality products is really, really important for us for both pest management and that resistance management piece. So when you start to lose those tools, you have that loss of flexibility. It's not just the loss of flexibility from your control, but also, you know, how do you work with your biocontrol agents when you're starting to lose some of those products? Also, we are a pretty small marketplace in Canada. I kind of meant to, and maybe we'll get a sense of this because I'm kind of assuming that there's a lot of people here in Canada, but maybe some people from the States as well. But we don't have as many active ingredients available for use as perhaps you do in the United States. And so that, you know, fewer modes of action mean we have fewer choices right from the get-go. Part of that is that we're a small market. We're not necessarily the first who are going to receive registrations. I mean, it's expensive to register a product. And actually, when you think about the cost of developing products from that whole discovery phase all the way to bringing a product to market, that cost is huge, and it can take well over a decade to actually occur. And then, of course, we have the issue of reevaluations and further reducing our pesticide options. So I think this is why I'm saying these, you know, pesticides are valuable, and we need to see them as non-renewable resources when we're using them. You've probably seen this picture before. If you've had Jason DeVoe, our application technology person, come and speak, where he's showing this water-sensitive paper with coverage, different levels of coverage. That's kind of the reminder for me to talk about resistance, and is it actually resistance that you're dealing with? So for example, if you are working with the right product, you know that it's the right product for that particular pest, the life stage is the life stage you want to target, you're using the right rate, you're using the right spray water pH, the right water volume, your calibration is awesome, and there are no other conditions that are maybe affecting performance. You know, there's always that question is the reason that what I just applied not working? And I know that comes through a lot. And unfortunately, we don't always know. We often don't know what the status of resistance is to a pest. And so that's kind of always in our mind. So I'm, again, kind of assuming that all of these other causes of poor control have been addressed when I'm giving this talk today. So then there's the whole piece about what is pesticide resistance, really. So, I mean, in basic terms, it's a selection process, and it occurs within a population's gene pool. So populations contain genetic diversity. So if you look on the slide here, there's a bunch of little caterpillars, and most of them are black, and there's a couple that are red, and a bunch of other colors there. And that's meant to represent that diversity. So there's, you know, mutations, and that's why they're different that are in there. And sometimes those mutations make things less sensitive to a given pesticide. So when individuals within that population are exposed to a pressure, like some kind of stressor, in this case, we're talking about insecticides, some are going to fare better than others, right? So resistance is a heritable trait, and that means that it gets passed down from one generation to the next. And in the cases of pesticides, susceptible individuals that are exposed to the pesticide, well, they may not survive to reproduce, unfortunately for them. But the resistant ones get to pass on their genes to the next generation. And then over time, if the selection pressure is maintained, everything else is equal. Numbers of resistant individuals have the potential to dominate in the population. And of course, that occurs at different rates. The speed at which that can actually occur depends on a bunch of factors, but in general, the more you use a mode of action, the more likely you are to drive something to resistant. And what we're really trying to do for resistance management is try to maintain those susceptible individuals in a population. It's really important to do that. And this is just graphically what this looks like. You can see on the leaf, there's a whole bunch of different insects that are there. Again, the black ones are the susceptible ones, and that's kind of the first point in time. And what happens here is as you frequently use the same mode of action repeatedly over time, maybe not leaving enough time between applications of that mode of action, what you can do is you can shift the population from being mostly susceptible to one that is mostly resistant. And that's when you're going to start to see problems occurring from a performance standpoint. So I certainly don't want to spend much time or really any time talking about how insecticides work. I mentioned that IRAC website. If you want a really great slide deck on that, there's one there that you can take a look at. But basically how insecticides and actually how pesticides work is they act on key proteins that are necessary for the survival of the insect. So really important functions within the insect that keep it alive. And those proteins are things like enzymes or receptors or channels within the insect. And they have different binding sites to them. So a couple of the terms that you'll hear when people are talking about resistance management is target site and mode of action. So target site is really the site of action. And that's the place that's on or within that protein where the pesticide is going to bind to. Whereas mode of action is that interaction of the pesticide with that action site. And so insecticides that work in the same way, they get grouped into the Insecticide Resistant Action Committee or IRAC's mode of action classification system, which is mode of action is denoted by numbers, and they do have subgroups that are noted by letters. So I have a picture there of the different modes of action that are effective on Lepidoptera. So you can see there's a bunch of different targeted areas in the physiology. So things like nerves and muscles and growth and development, respiration, which is like that energy production, midgut, protein suppressors, and then all these unknown and non-specific ones as well. But what you really need to know from a rotation standpoint that's really important are what are the numbers, because I will talk about this repeatedly in this presentation, but that rotation by the numbers is the approach that is generally recommended for insecticide resistance management. Just a quick little picture to show one of those little proteins. There's a target protein. In this case, it's a channel that is like an intracellular channel. And you can see there's a little binding pocket. And normally something else would bind there to actually do something so that that protein can actually function. But in this case, there's actually a little insecticide that will fit in that space as well. And when it binds to it, something happens that shouldn't happen, or nothing happens at all. A protein can have more than one binding pocket for these little molecules to it, and then those binding pockets can accommodate chemicals with different structures. So that's why, when you talk about things like these, the group 28 diamides, there's a bunch of different ingredients, and yes, they all work in the same that same target, but they may have slightly different binding. There's a bunch of different mechanisms for resistance. You've probably heard about things like behavioral resistance, where an insect, comes in contact with something and says, "I don't want to be there," and they leave. Or there can be penetration resistance, so that there's something about the cuticle that makes it harder for the pesticide to get in. And then there's some really cool stuff with gut or symbionts and stuff in the gut of the insect, the microbiome, which can affect how an insecticide works on an insect. But the two main ones that we usually talk about, because they're the most common, are both target site mechanisms of resistance and metabolic resistance. So look at target site, it's usually described as being like a lock and a key. So if you think about the target site as it's a lock, there's an insecticide that can fit in as a key. And if it does so, it really disrupts everything. But sometimes those target sites, there are mutations to them, and that mutation occurs, and then that insecticide no longer fits into that site. So if it can't bind to the site, it doesn't work anymore. So when that actually happens, it's a very, very specific resistance, but it does confer really high levels of insensitivity to that pesticide. Metabolic resistance is really quite problematic because it actually involves enhanced enzymes. So insects have a whole bunch of different enzymes that do things. And in some cases, there are mutations so that an insect will produce a lot more of a certain type of enzyme. And what these do is they will actually break down the pesticide before they actually even get to the target. So doesn't get to the target, doesn't work. And these enzymes can sometimes work against multiple insecticides as well. So this is the most common form of resistance, and it is really problematic. They both are. Pests can also become resistant to more than one insecticide. So we talk about cross resistance and multiple resistance. So cross resistance is resistance to 2 or more pesticides via a single mechanism. So it can occur within a mode of action class and sometimes between 2 or more mode of action classes. And a really good example here of how cross resistance can happen is where you have an enzyme that's able to detoxify different structurally, like insecticides that have different structures to them. So for example, if I look at the group 4A, which are neonicotinoids, there's a bunch of different neonics within that group. So I can have an insect that it's developed resistance to, let's say, imidacloprid, and it's via this metabolic process of an enzyme. And then you expose it to acetamiprid and it's never seen it before, but it's already, I mean, it's resistant to imidacloprid and it also happens to be resistant to uh, acetamiprid. So this is that concept of enzymes can break down more than one thing. And then there's the multiple resistance, where you can have 2 or more pesticide mode of actions that are affected by multiple mechanisms, whether they're target site or metabolic or both. And there's a bunch of factors that are affecting, the risk of resistance, and you can't control a lot of them. So you can't control how an insect reproduces, or the number of generations that it has, or the generation time. You don't have much control on how mobile the insect is, whether or not there are susceptible individuals coming into your production or not. And of course, that's important because they get to mate with the existing gene pool, some of which will be resistant. You can't control how sensitive or insensitive the insect is to that group of insecticides, and you don't have control over host range. And of course, insects that feed on lots of different plant types are exposed to and used to detoxifying a whole bunch of different types of plant defense compounds. You don't know you can't control the number of the genes or whether they're dominant or recessive. And you can't really control what that stability of resistance is either. So when we talk about what is the stability of resistance, a lot of insects, when they have a resistance, we kind of make this assumption that there are fitness costs associated with it. So yes, I'm resistant, but maybe I'm not as good at finding a mate, or I can't fly as far, or I can't lay as many eggs, or my eggs aren't as viable. There's a cost associated with having that resistance there. And when that happens, if the pressure of resistance is removed, the idea is that they're no longer at an advantage. And the susceptible individuals, if they're present in the population, can start to build up again. And that's actually, a pretty important thing. And that piece of knowledge is very important because it relates to something that we refer to as the frequency of resistance, because that is something that can change. And there are agronomic practices that you can do that will help this to occur. So let's take a look at that frequency of resistance. So you've got a population of insects, and there's a proportion of them, hopefully really small or not at all, that are resistant to a given mode of action. And because of that, those fitness costs, if those pressures are removed, you can have a shift from more resistant population to one that is more susceptible. So, I mean, from a greenhouse standpoint, you're often getting in source material, or you might have insects that are moving outside into your production areas. You don't know what the frequency of resistance is like at that source, right? You kind of have to assume that there might be resistance present, and you have to try to engage in practices that are going to help to move that in your favor, moving from a more resistant to a more susceptible one. So again, that frequency is something that you can work with. The other thing is the frequency and intensity of the resistance. So sometimes there's low intensity resistance that's present, like there is a level of resistance that's there, but you're still able to achieve relatively good levels of control. If you're using a product very occasionally as part of a resistance program, you might not even notice it. But if you've got a really high intensity of resistance, you might notice that very quickly. And so, that's a really unfortunate situation, because it really doesn't matter what you throw at a population where there's a high level of this sort of high frequency and a high intensity of resistance. So if you're going to go and throw 10 times the rate, not that anybody would do that, it's not only off label and illegal, but also probably is not going to be effective in the short or the long term. And we can kind of use that information here to, that whole idea about fitness costs and the frequency changing and shifting that, because you can take advantage of that unstable resistance to bring that frequency down by limiting the number of sprays that you're using of the same mode of action, then having that interval of time between use. So in the picture I have on the left-hand side, it shows the frequency of resistance after a spray. So, of course, there might be some individuals that have some level of resistance to a pesticide, and you spray your product and you kill a lot of the susceptible ones. Some of the resistant ones remain. But then again, over time, there's those fitness costs that are operating. Maybe you've got susceptibles moving in from elsewhere as well. And so that baseline level or frequency of resistance goes down. If you leave enough time between them and you're doing other things, you're using other products, you're resetting that to baseline. But what happens when you use a product too often, too frequently, not enough mode of actions being used, you can get this what we call a stair-step increase in resistance frequency. So that's really what we want to avoid. So there's really these three generic resistance management strategies that are talked about, and that's moderation, multiple attack, and saturation. And moderation and they're not necessarily mutually exclusive either. So moderation is exactly what it sounds like. You are reducing the overall use of insecticides overall, and certainly of specific ones over and over again. And that's reducing the exposure level in the population and helping to preserve those susceptibles. Then there's multiple attack, which is the idea of diversity. So using multiple different modes of action, and actually rotations are a form of that moderation, because you're using the specific mode of action less often. I also mentioned mixtures because it is often talked about from a multiple attack point. And you'll see it perhaps more frequently in the fungicide world. But for various different reasons, including the fact that we have new tank mix guidelines that came into effect last year, mixtures are something that we don't necessarily have as options, but also are not necessarily what are recommended from a resistance management strategy. I know sometimes mixing two things together can work really well, but that's actually not the approach that is generally recommended for insecticides. And then there's the whole idea about saturation and trying to basically overcome resistance. And the idea here is just, you're using really high rates to try to eliminate all of the resistant individuals. But in practice, this really does not work, and it tends to end up selecting for resistant traits. And I'm not talking about label rates, I'm talking about over label rates here. So that's sort of the background on that. So what are some tactics that we can implement in the greenhouse? And I, before things get really bad, you don't want things to get really bad. You want to have a plan in place ahead of time. So you know what crops you're producing and what varieties. And yeah, if you're looking at some of the material that comes out on floriculture and on vegetables, there's some really great information there that both Sarah and Kara have provided on relative susceptibility of things. You might know what the pest pressure is like based on previous seasons. You probably have a pretty good idea of the pest complex. I know that there are surprise visitors, and that always makes things a little bit more complicated, but you can do a lot of planning ahead of time. if you can find out what the history is for your incoming plant material, like if you are getting material in from areas where you know that there may be some resistance issues, then that planning ahead is really important. You may not know the specifics, but you may have some ideas. This is where we get back to this whole non-chemical tactics wherever possible, all of the other practices that you need to be doing as part of an IPM program. And when I say non-chemical tactics, I am suggesting that you can use microbial pesticides here. I don't want to exclude them. If you can use non-selective products like soaps and oils, these are fantastic. They are not prone to resistance, and these are things that provide either mechanical or physical disruption of the insect. And don't apply pesticides if you don't have to, right? I mean, this is based on monitoring and thresholds, whether it's a numbers of insects or whether it's some kind of damage rating that you're using. Those treatment thresholds are really important. And you need to decide ahead of time, like when are you going to actually intervene? So having those intervention decision deadlines are critically important. And if you can delay applications of pesticides longer, and I say this because our programs, our IPM programs for greenhouse production are very, very much tied to biocontrol, the use of biocontrol, and it's because of resistance that has occurred in the past. And we don't have as many options. So we want to make sure that every application counts. And evaluate your interventions, right? Like if you can apply something ahead of time, get an idea of whether it's going to work, maybe a small, very, very small portion of your range, that's great. And certainly after you've made an application to part of, or well, hopefully not all of your range if you don't have to, but evaluate your interventions. Did things actually work? Or is there some indication that maybe there's a level of insensitivity to the chemistry you just tested? One of the things I've seen recently that Sarah has been talking about, but I know it's based on an awful lot of work that's gone on with the Vineland Research and Innovation Center, is the whole idea about having dips ahead of time to help start clean. So there's always a possibility that things are coming in. You may not see them. You don't usually have adults that are present on things, but sometimes there's immatures that are lurking in plant material. And so dips can be incredibly effective at knocking those numbers down or eliminating them. And that really does help you to start clean right from the get-go. And it can really delay the need for insecticides and preserve those critical uses for later in the production cycle when you really need them, like especially towards the end where you absolutely need to have a clean crop. There are several great biorational products registered for use in Canada. The ones that I've listed here, I think Sarah updated this for me, but basically you have some Botanigard, Bioceres, soaps, and landscape oils available at your disposal for thrips, whitefly, and aphids, which is great. They are lower risk from a resistance management standpoint, and they can be really effective in terms of buying time before using other products and or between uses of, I guess, your A-team type products. So I love this. I love this slide here. It is stolen from one of Sarah's presentations. I know she's talked about it. And I before. I think what I like about it is actually quite an elegant slide when you think about it. I mean, there's an awful lot that's going on here. Obviously, we're talking about a crop that is long-term crop, right? It's definitely not of short duration. So there's a lot of time where you have to actually think about what am I going to do? And here, the concept of starting clean is evident, right? I mean, you're dipping your cuttings, you're dipping your rooted cuttings before potting when you get them in. You're using biocontrol and mass trapping as the core, as the foundation of your pest management program. And then, if you're doing your assessments, you're monitoring and you're over threshold and you have to go in with a pesticide, then you try to delay that as long as possible. Reassess how things are going, ongoing monitoring, reintroduce your bios if you can, and then that cleanup that happens before sale. So there's a lot going on here, and it looks simple, right? But I think it's many, many years in the making to try to make a program like this actually work. So recognizing it's not an easy thing to do. So one of the other things, and this is part of it too, is the whole, do you have to spray the entire range, or can you spot treat? If you know that the crops that you're working with, like some of them are more sensitive than others, or even some varieties or colors are more sensitive than others, you may be able to treat those hot spots instead of doing the entire range. And this is really important, again, from a resistance management standpoint, because it's leaving a safe space for those susceptible individuals. You want to keep those susceptibles in the greenhouse because you know there's going to be individuals there, so they may as well be susceptible and easier to manage later down the road. And it's hard to do that in a greenhouse when you think about it. Certainly in the field, what we do is, if you leave some areas that are untreated. So I don't work in like the big field crops, where we talk about having, mosaics, or whether you talking refuges and things like that. But it's the same idea. You have areas where the pest isn't being treated. It's hanging out on its own. It's not being sprayed, and it will eventually move into the cropping situation, or vice versa, depending on how the pest overwinters and moves around. That movement and mating occurs to mix things up. And the way to do that in the greenhouse, if you can, is really just to treat those hotspots. You need to be able to keep track of the pesticides that you're using. So when did you use, where did you use them in the production cycle? And again, track their performance just to see, do you need to retreat or not? Or, is there some kind of change of sensitivity? I think that's important. Can you get away with suppression? Do you have to use the A-team type most effective products all the time, or is a suppression tool going to suffice? Because sometimes it's good enough, certainly great as a way to rotate to something different from a resistance management standpoint. Like recognizing, of course, you need to be able to do things like cleanup before the sale of things at the end of the production. You want to have good products available at that time if you need them. The other thing to think about, I think, is that whole persistence of the product that you're using. I mean, persistent products can be great on one hand, but on the other hand, they tend to have some unintended consequences. I'm sure that there's been presentations over time on things like hormesis. So it's kind of that stimulatory effect on the insect that can affect things like how many eggs it lays from sublethal doses. So not enough to kill something, not enough to really necessarily even make it sick, but it is enough of a stressor that it can stimulate other aspects of the insect's life. There was an interesting article that came out a few years back on the impact of fungicides and how they influence the fitness of Colorado potato beetle for later exposure to insecticides, and how that affected their susceptibility to them. Pretty interesting stuff. The same kind of idea here with those sublethal doses or just exposure to a stressor. And then, of course, the problem with persistent products is it has a much more negative effect on susceptible individuals. They tend to be suppressed for longer periods of time. So if we look at things like that whole idea of persistence, what's actually happening with these long residual products is what they tend to do is they kill the susceptible, or they keep the susceptible ones out and dead for much longer than they affect the resistance ones. And so it takes a very, very long time before you start to see that reset occurring back to that baseline. And of course, there can be impacts to your bios that are in there as well that may be affected by sublethal doses as well. And it varies. And it's hard to determine how long that residual activity actually occurs over. I want to stress the importance of using label rates for products. I know sometimes there are label rate ranges, when I say using the label rate range, so if I talk about underdosing or overdosing, you're outside of the label rate range. If you are underdosing, it can be a real problem. Like going under the label rate, it can really accelerate resistance. Again, it's that whole idea of you actually end up killing susceptible individuals in the population, and you're allowing individuals with low levels of resistance to survive and to reproduce. It is a problem. And I know that there has been some I've heard some chatter about, well, if I use more frequent applications of these really low doses, maybe that's good from a pest management and resistance management practice, but it actually isn't, because you're killing those susceptibles. You also don't want to be used higher than label rates. Obviously, it's off-label and very expensive. But the issue is that we know that when you start blasting insects with really high rates, especially repeatedly, that's actually how you accelerate resistance in the lab, and that is likely what is actually happening in the field as well. So again, the whole idea here is you really do want to keep those susceptibles in the population. So how do we, how do we rotate? Like, how do we put this into practice? For insecticides, the recommendation is to rotate by the numbers and to use treatment windows. So a treatment window is basically defined as a generation in this case. And a generation, obviously, it varies. If you can monitor a population, like I can do in the field, for example, let's say oriental fruit moth in an orchard situation, I might have 3 generations. They're pretty discrete. I mean, I can monitor with them with pheromone traps. I've got degree day models, so I know when to apply my products. They don't overlap the generations at all. And the idea here is that each generation gets treated with this, like the same mode of action, and then I switch to a different mode of action for the next generation. So I might need more than one spray to control oriental fruit moth in a specific generation, but I choose a pesticide with the same mode of action, and then I switch the next time. It gets a little bit harder for short generation pests because they often have these multiple, like short overlapping, right? You've got overlapping life stages. So you've got all life stages present, and it gets really, really messy to do, recognizing that for sure. So in those situations, what IRAC normally does recommend for windows is that they will say for short generation pests like aphids or mites, use 2 weeks. If it's something like whitefly, it's 30 days. So that's kind of how they base that. So how would you actually, what does this actually look like? This is a really simplified example because it involves one pest that has 2 generations per year. So what you really don't want to be doing, obviously, is using the same product all the time, right? There's no rotation there. There's a very high selection pressure that's occurring, especially if you're in an area where there isn't a lot of movement of the pest. The second the second option that they talk about is that rotating within a generation. So I apply one pesticide and then I apply a different mode of action for the next, like within the same generation, but this second spray that I need. And then I do the same thing in that second generation. You don't really want to be doing that. The reason is you are exposing 2 generations to the same chemistries. And that's a risk to both of the mode of actions. What is often recommended, especially if you don't have a lot of products to work with, is that whole Windows-based approach where I'm treating the first generation with the one mode of action and the second generation with a different mode of action. That is actually a pretty good strategy because it does allow some recovery of susceptible populations to occur. Now, if you're really lucky and you have a ton of products at your disposal, which you probably don't, there is an option to kind of do that whole, I change my product with every spray, but it's not really impractical. It's not really practical. It can be very expensive to have that many chemistries. And there probably in most situations isn't a huge advantage to doing so. There's some great strategies on IRAC that talk about some of the pests that you deal with in greenhouse situations. One of them is for Bemisia. And you'll see here on this poster, which you can download or I can send it to you. They do talk about some of the main resistance mechanisms that have been seen. They talk about different chemical mode of actions that work against this pest. But more importantly, you'll see that whole mode of action treatment approach, which is based on that changing it up for each generation. So each 30-day interval gets a different mode of action. And then something similar has been done for tomato leaf miner, which I know this is a terrifying pest. I'm not going to talk about it much. It scares me just thinking about it. But it's the same kind of thing where they do talk about having these intervals. In this case, they've determined these 30-day intervals, and they do switch it up. Now, they do have combinations of products that are going to be applied there. In this particular rotation example, but what is important is that there's no back-to-back use of the same mode of action. So what about tank mixtures for resistance management? I know that this is something that is sometimes recommended, but from our standpoint here, I mean, first of all, it's the tank mix guidelines that came out, I think, December of last year. They are posted online. There are some restrictions, so you really need to be able to look at what each of the labels say that you're planning on mixing together to find out whether that's even a possibility. Then there's always the question of, is the mix, like, is it biologically, chemically, or physically compatible? Because it may not actually be. You may even have some antagonistic effects from a pest management performance. They can broaden the pest spectrum. So, if you're throwing more than one thing in the tank, the idea being I've got multiple pests there, I need to control them, and I need different types of products. I think that's probably valid, but it is not a resistance management recommendation as a primary focus. When you start using mixtures too, you're also limiting the future product choices that you have in the crop cycle. And again, we don't have a ton of stuff here. It makes it problematic. And if it would still require rotations of mixtures. So on that slide with Tuda, where they had some, multiple mode of actions being used, it looked like they might have been tank mix. I'm not positive. But the reality is that you still need to be able to rotate them. You don't want back-to-back generations exposed to the same things. So here's another one of these really nice, elegant slides from one of Sarah's presentations where she's talking about parvy management. And I think it's Mandevilla. Hope it is. But what I love about this and why it is really elegant is it brings in so many of the concepts for resistance management. I mean, first of all, there's that whole, you're monitoring. You've got a number in mind, a threshold in mind that's been established, and you're working with that. You're using your bios and other forms of control early on. And if you have to go in with insecticides, you do so. Being careful with what you're choosing, obviously. But what you'll notice with this slide is she's got 6 different mode of actions through this activity period, right? Like there's a bunch of them. And also just things like there's different varietal susceptibility to the pest. So, you don't necessarily have to go in as aggressively on one variety as another. So all of those kinds of things are in this slide. And I think this is why it makes it a really elegant slide and just a lot of great information that's in there. And another part of that is that whole rotation. Yes, you have products that you're reaching for because they work really well against adults and larvae. And when you need to have something that's super, super effective and the timing is right for them, that's great. But you need to have rotation partners for them. And that's where the B team comes in. Some of them may be suppression only, but they are really important for resistance management, and they can really, really add to that program. One of the things too is these examples here are pesticides for Parvis spinus, right? But there's other pests that you're dealing with in the system, and you always have to think about what else am I exposing these pesticides to. So how do you build an IRM program? Again, there's some really great examples on IRAC online. I find they're quite simple, like they're really simplified, and you can use them as ideas and the foundation for building your own program. But here in this case here, we've got whiteflies and we've got these 3 application windows that are 30 days in duration. Now we know, of course, that reality check is that when it's super hot outside, the development, the generation time can be really fast. And by generation time, I mean the time it takes to go from an egg to a reproducing adult, right? So in this case here, they have it as 30 days, but it might be less than that. Which is something that you would have to adjust to. But what's important there is, there's multiple applications that are required in each window. They're using the same mode of action in each window, and then they're switching to the next different mode of action for the next window. You'll see here too is there's multiple life stages present, right? I mean, you've got all the different life stages that are overlapping. But when you're using this Windows approach and you are switching it up, it really means that no matter what the life stage, the next generation is going to be exposed to a different mode of action. Again, so that's that rotation of mode of action. And then there's the whole idea about these overlapping pests. So in this scenario, and I'm so sorry that I've got corn here and not a beautiful greenhouse crop, but they don't have those in the IREX slide decks. I think they need to, but they don't. But what you have here is you've got 2 different pests. I'm not sure if it's supposed to be a beetle or not, but I'm going to assume it's a beetle and a caterpillar pest. And these little beetles, which seems unlikely that they would have such a short window of activity, but they have 15 days. It's 15 days between to go from one, like an egg, to a reproducing adult. So there's 3 little windows there. And then you've got this other pest that it takes longer to develop. It takes 30 days to go from an egg to a reproducing adult. What happens when you have those those sort of different life stage durations when they're superimposed on each other? You have to use the window that is shorter. So I'm basing my application mode of action rotations based on that shorter duration window period. And again, what you're seeing when you look at this slide, the reason it's strongly recommended is because you are rotating between those mode of action. So no one generation is exposed successively to the same one. So, and also note that that 4th window, that little green guy, it's not present. You're not actually targeting it at all. So again, just a reminder about monitoring and only applying for pests that are actually there. And then I wanted to try to build something here that's maybe a little bit more realistic for short crops. So there was a slide in the deck that IREC has put together that I think is kind of similar. And here it's production of lettuce, these short cycle crops. And here you have, let's say in this scenario, you have something where a transplant is being produced and then it's moved into a different area. You can see that when it's moving to it's probably supposed to be glasshouse and field, but when it's moving from the glasshouse to the other production area, you are switching your mode of action. So you're starting out with Y and then moving to X. You would, and then it has a second, I guess there's a second crop there, second planting, and they've changed it again. So they're starting out again with Z and then moving back to Y. So there's no back-to-back exposure of the pest in the production system at all that gets that back-to-back. Just a few comments on biopesticides and resistance management. Sometimes we'll hear things like they don't become resistant to things. And it may be true that it's less likely to occur. They can happen. Sometimes they're very complex and it's very unlikely to. It really depends on what type of bioinsecticide you're working to. But I mean, there are examples of the development of resistance to, let's say, granulosis viruses, and certainly for BT and things like that. So these are possible. We do need to think about them, but they're very, very useful tools. And they're great rotation products. This is just an example of the different types of products that are out there. And you can see there's some synthetic pesticides as well as bacteria, fungi, things like that. And they talk about whether or not there's been field resistance, evidence of it in the lab. So you can see for some of these compounds, it's pretty slim to none, but it's not impossible. Reality is pretty messy when you're trying to put together a program like this. I mean, we talk about windows and application windows, and then there's a wrench thrown in saying, well, if you've got a pesticide that has a long residual activity, then you need to use the duration of the residual effect as your window. Well, how do you know with the residual effect how long it actually is? I mean, does it mean how long it's effective against the pest? Does it mean what's the residual activity against others, against your bios? It's a hard thing to work around. And of course, systemic products can have that extended residual activity. So it's really hard to do. And then, of course, you're trying to consider your bios, right? You want to choose products that are compatible wherever you can. But you certainly don't want to overuse a safe mode of action just because it's compatible, because you might lose it in the end. Existing resistance, if it's occurring, you're going to have to switch to other tactics and maybe focus again more on some of those non-chemical tactics. You might, if you're worried, seeing shifts in sensitivity, you should be changing to other things. So if you suspect resistance, stop using the chemical in question and others within the same group mode of action, because applying higher rates more frequently is not going to help you over time, been some cool work with two-spotted spider mite that's been done by Western University in Ag Canada. I think you've probably had some presentations by the Vava Gerbic in the past. I mentioned the IRAC website, fantastic resources that are there. Please visit it, just some great information. So just to recap, plan ahead wherever you can. Start clean and use multiple tactics. Base your interventions on monitoring and keep a safe space for your susceptibles and for your bios. Think about the life stage, the rates, the pest complex, the persistence of the products that you're using. Make sure to rotate between the mode of actions. Don't forget about soap, soils, fungal agents. Evaluate and talk to each other. And that's the end of my talk. So I am going to stop sharing.
Thanks so much, Hannah. That was really great. If anyone has questions, reminder to put them in the Q&A little button. I know this information can be quite technical and a little bit intimidating, but remember, there's no dumb questions, only dumb answers from us. I had a few questions come at me directly from the chat, though, so we can address a couple of those. One of the things is you gave that, I mean, there was lots of great IRAC examples of the pesticide spray windows. And I noticed, and one of our viewers noticed that one of the recommendations was to spray the same mode of action twice in a window and then move on. Why is it 2 and not 3 or 4 or 5? What is there a reasoning behind that?
Yeah. And always, of course, and I just want to say, you have to look at the label, right? Because the label has information on how many applications too, right? Just always say that. It's 2 is usually like, okay, assuming you need to apply more than once, like that's the assumption is you've gone and you've sprayed. Yes, you know, hopefully it's the only time you have to spray for it, but that's, you know, often not realistic, especially when you've got those multiple life stages, right? 2 is recommended instead of going for that third one. It's that additional level of pressure. It may not be valid for every pest. It kind of like, especially when I start thinking about some of the field pests that are there, you think, well, why couldn't I do a third application of it if it's still within that window? But it just, I mean, are you talking about sustained? If you're working within the window of the pest, I'm trying to explain this properly. If you're working within the window of the pest, chances are you're going to get away with 2 sprays and not 3. I'm not sure what kind of situation you would need 3 for. I guess that's my question back to that. So Sarah, when would you need 3 within, let's say, let's say within a 30-day period?
I guess an insect with a really short life development cycle, yeah.
But then the window's shorter, right?
Yeah.
If you're dealing with a pest that has a 2-week interval, that 2-week interval, probably 2 sprays is all you're going to be able to get in there anyway.
Yeah, good point. One of the questions that just came through the chat is someone was asking, are there resources for actually determining what the residual amount of pesticides would be? And I know we can do some sort of pesticide testing, but I also know it's expensive, and I think only private labs do it. Is that correct?
Yeah, I actually have tried to find that because I get that question. I've had the request of, can you make a list of all the different active ingredients and the different products and formulations and say, you know, how do they work? Okay, how they work, that's fine. Are they contact ingestion? Yeah, no problem. Are they translaminar or, you know, fully systemic or contact? Yeah, no problem. Can you tell me how long they work for? No. I really can't. But it is a really good question. I mean, there's some information on the label that should give you an idea from an efficacy standpoint. So sometimes it'll say like, you can retreat in 7 days, right? Or retreat 5 to 7 days later, or retreat at 14-day intervals. And that kind of gives the idea, gives you an idea of residual activity, right? It kind of gives you an idea of that. But it's not a guarantee, and I don't have that information. It is of course it's incredibly desirable. And then there's the whole, well, how long are there, like how persistent is it? Is it 1 week or is it 6 weeks? Like how long is my bio going to be affected by the spray that I just used? And a lot of that, I think, probably comes down to your experience. And it's going to be different in a greenhouse than in a field situation too, right? How quickly things break down. Then there's also the whole thing is like, what was the coverage like, right? What was the coverage like for it?
Yeah,
So
and
there's a lot of things.
if it's systemic, sometimes the plant
Exactly.
start to put out new growth, which then might not have the systemic chemical in
Yeah,
it.
depending
So
on
that
what it
might be-
is. Yeah,
Yeah.
it is a really good question, and I would love to give you that as an answer. And I did try to find some examples, but I can't.
I think that's where scouting comes
It
in,
is.
and
Yeah,
flagging plants, that maybe had aphids, for example, before you spraying it and then see if they
yeah,
come back in that
and how
surrounding
quickly.
area. Yeah,
Yeah,
so sometimes just monitoring
absolutely.
can tell you that.
Yeah,
And then if you are recording that and having spray records and what worked and what didn't and how long it lasted on your own farm, you might be able to answer that because there's a lot of components, especially in floriculture when we grow so
yeah,
much different stuff.
yeah. It's a really good
Um,
question.
yeah. Someone's asking a very good question, but a very complicated question that I don't know if any of us can really answer. But in a program that uses both biocontrol agents and chemicals, is there a certain order you would suggest applying them in? And what does this depend on? And it doesn't matter if you're using biocontrol friendly chemicals or not.
Well, I mean, I think from our standpoint in Ontario, like in Canada, we probably reach for bios first. Like, I mean, clean up with those again, the soaps and oils. If you can do that kind of thing, that's great. Or some of the other bioinsecticides, great. But then, I mean, eventually you may get to a point where you have to apply a product. And I guess part of it depends on where you are in the cycle, what part of the plant is at risk, what your pest pressure is like. And whether you actually want to consider going back to using a bio, right? Because, there are some products that if you're going to use them, then it's going to be a while before you can go back in with the bio, or maybe not at all, depending on the crop, right?
Yeah, a lot of
I
consideration.
don't know if that's what you're asking, but I mean that's
No,
kind
I
of
think
I
that
think
is,
that's the
yeah,
approach, right?
yeah, and again, I think it matters by crop and how short versus long the crop is and
Yeah.
all of that stuff. So yeah, like I said, a great question, but an intricate one, especially for what we do in greenhouses.
And actually, like that is one of the things. Like I don't know how many people who are producing crops in the field on the call today, but when I started thinking about this talk and realizing, oh, it's just resistance management strategies. It's not really like it's not really complicated. But then when you start looking at greenness, it's like it's so complicated. It's a lot more complicated. So I mean, I think that these kinds of discussions that we have in these workshops, and they're really valuable in that sharing of information. Yeah, and how to make it work.
Another question that just came over the chat is, can bios become resistant to insecticides?
Sure, they
Biocontrol
can.
agents? Yeah.
They can be. I mean, I'm always my experience in thinking about that has come from these perennial systems that occur outdoors, right? So, I mean, this is like years ago, but I don't think people have really studied it recently. There were uh, there were some issues with loss of predatory mites because of the programs people were using for pests like oriental fruit moth and codling moth. And that was having, of course, issues because now suddenly European red mites were problems in orchards, right? It's like, oh my gosh, right? Because of course there's this cascading effect. But what they were finding eventually is that there was resistance developing in those bios, but they had time to develop that resistance. And I don't know in a greenhouse setting whether that time there would be enough time for that actually to occur. And I'm not talking about releasing resistant bios. I'm talking about that selection process that's occurring on the bios as it is. Plus, it depends on the bio. Does it have a short life cycle as well, or does it take longer to develop? How vulnerable it is, and when is it vulnerable to the insecticide that it's being exposed to? Because not all life stages are as sensitive, and sometimes you'll have one life stage that's resistant to a pesticide, but the other isn't. Another one isn't. So it's complicated, but it's a very good question.
Yeah, I think back in the day there was some research
Yep.
into making pesticide resistant bios and then releasing
Yep.
those. I
Yep.
think maybe with a mite, but I don't think, yeah, if someone's mentioning Don
Yeah,
Elliott worked on
phalaensis.
that.
Yeah, that's
Places,
great.
yeah.
Yeah,
I don't know,
totally
whatever.
thinking.
Thanks, Mike. I knew it was like on the tip of my tongue and I couldn't quite get there. I don't know whatever happened to that, but it's too bad because that would have been exciting. But just moving on for a second, because there's a lot of questions coming in. This is a good one. If you suspect that some kind of resistance has been built by a certain chemical, how long should you abstain from using it? And then how can you check if resistance is still present after new generations? And I'll let Hannah answer this, and then I might be able to tack on a little bit to that.
Yeah. I mean, I think testing a small portion, like if you can set aside some plants and say, okay, I want to test it, right? What you're doing when you're testing it at label rates, like first I'll start at label rates, is that you're testing how sensitive it is, and you're looking for changes in sensitivity. So let's say, and by the way too, like if you've cleaned out your greenhouse, theoretically you could maybe be starting fresh. But let's say you're within your production, and it's a long cycle crop, and you had some issues with performance early on, and you for some reason want to try that chemistry again because you have it and you want to use it. I don't know. Try it in a small space, like a small portion, and see if it's actually working. How long? It depends on, and we was talking about that whole stability and fitness costs and things like that. I don't know how long it would take, and I wouldn't want to generalize. I don't think you can. I think you just have to try it if you
Yeah, I
must.
agree. I mean, I guess I
Yeah.
would wait a minimum 3 or 4
Yeah.
generations at the minimum. That's
Yeah.
not a rule. And I've seen lots of growers do really great Tupperware testing containers where you
Yeah.
sort of, tap some thrips or
Mhm.
what have you in a Tupperware container, put in some food so they don't just die, and spray, like just with a little spritzer.
Yeah.
The label rate, the important part of that component is to have a control
Yes.
to make sure they're not just dying from being left in a greenhouse in the Tupperware container or something like
Yep.
that. And yeah, just keep doing that until you get some dead ones. We've done experiments like that with Parvis spinus when we suspected resistance on Mandevilla. We were able to put some plants in some bug dorms and then spray different rates and different products at different times. And eventually we were able to figure out, okay, the resistance seems to have worn off, and go it back into the crop. So I highly recommend bug dorms and little experimental cages and all that. I guess I'll answer one last question and then maybe a comment. Someone is asking if you've got more information of European red mites in orchards. Maybe we can send out, Kara, if maybe you could
Yeah.
put
Yeah. Yeah.
Hannah's
Please do.
email in
Yeah.
the chat. That would be great.
Sure.
And you can email Hannah directly on that one. Someone was asking, can insects or mites develop resistant to oils? You mentioned microbial pesticides, but what about oils? And an example that was given was two-spotted spider mite in tomatoes. Sometimes growers seem to feel like it stops working at a certain point. Is that resistance, or is that some other mechanism maybe?
I don't think I've heard about resistance to oils or soaps.
Me neither, because I think
Um,
it
I,
suffocates them,
I
or yeah.
really, I mean, I can see reasons why things might not work, um, but I don't think it's a resistance. It's probably related to timing and exposure and life stage vulnerability, or
Yeah.
maybe even an accident made with how things were mixed
Yeah.
and the rate.
Yeah, and one comment or discussion point we had was that maybe, we're spraying more and running into resistance more because some of our biocontrol agents aren't maybe not as working as well as they used to. And are people aware of that? And I would just comment then when I went to the International Organization of Biocontrol meeting last summer at this time, and I can say that the biocontrol companies are aware of some of our concerns and are trying to address them through breeding and genetics and things like that. So hopefully in the future, things might turn around a little bit more. And I think we'll just have to end it there because I think we could spend all day answering questions about individual products or pests or rotation programs. If you've got any questions like that, feel free to reach out to Kara and I, and we can connect with Hannah and maybe answer some more specific or in-depth questions. And with that, I'll hand it back to Kara to lead us out and maybe where to find more information.
Great, thank you. So my virtual background didn't load this time, so you can see my my insect poster back there. Anyways, thanks everybody. Hannah, that was so great. We really appreciate you coming to talk about resistance management, because I feel like Sarah and I are both always telling people to practice resistance management, but to like provide us with a deeper understanding of how it works and why it's important and different things to do is really great. So thank you so much.
I had fun.
Good, awesome. We love having you in the greenhouse world. Anyways, we will post the recording to YouTube channels, and you can find them on our blog. So it's on floriculture.com and on vegetables.ca. We will also email, hopefully, a PDF version of this presentation, Hannah. Does that sound good?
Sure.
To participants, and hopefully you've got our contacts. I did type Hannah's email address in the chat, but didn't hit enter. And yeah, thanks so much. And we hope you'll join us again. Oh, there's also an exit survey. We would really appreciate it if you would fill that out. It really helps us kind of figure out if we're on the right track with information and things like that. Okay, so have a great day. Thanks again, Hannah, and we'll talk to you soon.